Hi everyone. Okay, we're going to solve this question using those kind of those steps that we set up for you in the note package there and that. And I want to get you really used to kind of using those steps, and they're pretty easy for these type of monohybrid questions. But when we start getting into more complex ones, if you follow the steps, it's just going to make that process so much easier for you. So we're going to follow that. So we know step one is to do a legend. So let's start reading and see if we can come up with some information for our legend. So it says, inflated pea pods are dominant C over constricted pea pods with a little c. So before you even move on to the rest of the question, we have enough information to start a legend. So that's what we're going to start. So step one, we said, is a legend. So we know that inflated pea pods are a capital C. So if I have a capital C, I don't care what this other allele is, and I like to put a line in there to indicate I know there's another allele there, but I don't really care what it is. As long as there's a capital C, this plant is going to be inflated or have these inflated pea pods. Okay? And constricted, the only way I can get constricted is if I have two little c's, because it is homozygous recessive. It's a recessive trait. So this is going to be constricted. Okay, and there's our legend. And these legends often are the most difficult, maybe not for these monohybrids, but like I say, when we get into more progressively more difficult type of questions, dihybrids and all sorts of questions like this, having a legend is, take your time on this. You really want to make sure you've done this part right. Okay, and then we know step two is underlying the parents in the question. Tell me their genotype. So we're going to read a little bit more. It says, use a Punnett square to determine the genotype and the phenotypes of a cross between a plant that is homozygous dominant, there's one of our parents, and cross that with a plant that is homozygous recessive. So again, step two, underline the parents. Let's say what the genotypes are. And often I just write it right above. So these problem-solving skills are really going to make these type of questions much easier if you follow the steps. So homozygous dom, we know what's dom just by our legend, and refer to your legend, that's why you've done it. Inflated is dominant. If it's homo, we know homo means same. Both those alleles, and again, genotypes are always in pairs, so both those alleles are going to be dominant C. So capital C, capital C. We're crossing out with a plant that's homozygous recessive. So we look at our recessive, we know that constricted is recessive, and the only way to get recessive is if I have two little recessive alleles. Okay, so that is step two. Step three, Punnett square. Okay, so we're going to have like a game of X's and O's. That's what you're going to kind of set up. And we just blank this guy out. Now, normally you have female on top and the male down the side. But in this case, we don't know who male or female is. So we're just going to isolate these genotypes that we underlined, and we're going to isolate them into gametes. That's what a Punnett square is doing for you. So let's just take this one here, and we're going to isolate it down to the side. We're going to make that a gamete. We're going to take the other one, and we're going to put it down here and make that a gamete. So these are actually gametes. This was a male. These we found these alleles within the sperm, right? Within the nucleus of the sperm that contains the chromosomes with these genes. We're going to do the same thing for, in this case, let's just say this is the female. Now we could have did it the other way arbitrarily. It doesn't matter because they don't, again, indicate which one's male or female. But if they did, we'd have to put the female on top. So same thing. We're going to isolate that into the top part of the Punnett square. Now you notice I put a little hook on these C's. Got to be careful if you're using letters that kind of look the same from the capital to the small or to the smaller case. And if it's just size, that's when we start doing these more complex dihybrid 16 Punnett squares, often you get mixed up what's big, what's small. So make your little guys with a little hook or a little line over them, but really distinguish them from the capital letter. Otherwise, you're going to have problems when it comes to interpretation. Okay, so that's what we did. That is step three, Punnett square, isolating the genotypes into individual gametes. Okay, Now, step 4, we're just going to bring these guys together. So we're going to bring the gametes together to simulate fertilization. And the rule is when you're doing the genotypes of the offspring, and that's what we're doing here in these blocks, you have to show the dominant allele first. Okay, and it makes interpretation much easier later. So capital C, little c. Okay, and we do that for all the rest of the offspring here. Capital C, and then bring that one down, little c. Now, the other thing we want to do is when we're filling in, so that's step 4, we're filling in the Punnett square right now. Inside the little block, give yourself enough room to actually put the phenotype as well. And in the end, when we're going to count up and we're going to look at phenotypic ratios or probability or anything like that, it's much easier to take a look at the phenotypes if you put them in each of the blocks. Easier to count those guys than it is to look at the genotypes. So let's go back up here. We look at our legend. As long as I have a capital C, I don't care what this other allele is. This is going to be inflated. Okay, and this one also inflated. Okay, do that for the rest. Capital C, bring this guy down, little c, that's going to be inflated. And then capital C, little c. This one's going to be inflated. So we count them all up. 1, 2, 4 out of 4 boxes is inflated. If we want to do if they ask it for a fraction, 4 out of 4 would be reduced. And remember, you must reduce your fractions. That would just be 1 out of 1. Okay, which is would be, of course, inflated. If they ask for a phenotypic ratio, it would be 1 or 100% are going to be inflated. And often what they'll do is they'll you can put that just as a 1 and there's nothing else here for the rest of the ratio. So that would work there. So on a test, on an exam, they'll be very specific on what they want the answer in, whether it's a fraction, whether it's a decimal, a percentage, or just a phenotypic ratio. Okay, if you have any issues with this, guys, give me an email and we can go over it together. Okay, thanks, guys. Bye.